So today we will be talking about the antibody. Now we all know antibodies are nothing but the antigen binding antibody, something which is binding to the bodies which are coming from the outside and trying to harm. That's why it is called as antibody. So antibody are the glycoproteins which are present on the B cell membrane, or it can be secreted by the plasma cells in the plasma or the tissue fluid. When we talk about the antibody, the major function of the antibody is to bind to the antigen which stimulated its production, as well as antibody calls the other molecule means cellular material to kill that things. So generally when we talk about antibody, so antibody we had already learned that it is prepared by the antigen stimulation by the B cells. Now B cells have two types of antibody. One is the membrane bound. Membrane bound means this B cells will have the antibody in the form of receptor on their surface molecule, as well as when the B cell differentiated, what we have learned into the this humoral immune response, that B cell, when it gets the secondary response, it differentiates into the plasma cells and start secreting the antibody. So these secreted antibodies are nothing but these are the plasma soluble material. That's why we are telling it as a humoral immune response, because the term humoral means something which is soluble into the fluid. So this antibody is having two important jobs. The first job, to kill the antigen who has stimulated the production, because we know antibodies are very specific. They are formed only and only when the immune system is challenged by the antigen, and then they are getting the stimulation from the Th cells that this antigen should be caught up and to be cleaned up by the antibody. So first thing, it kills or it binds, locks the antigen for what it has been formed. And the second important job, it not only logs that particular antigen, but also it has ability to recruit the other immune cells. That means, what do we mean by recruitment of the other immune cells? Recruitment of other immune cells means sending the chemotactic signals which gives stimulation to macrophages and other type of immune cells which we have learned in the detail. So all these cells have been called, and these cells are taking care of that antigen. That means the various other immune responses like complement acting system, cytokinesis, everything happens after that. Now we will learn in detail about the structure. Basically, this particular lecture we will be talking more and more about the antibody structure. So antibody, when we see generally, it is the globulin protein, and it reacts specifically with the antigen that we had already discussed, which has stimulated production of that particular antigen. So when we talk about the antibodies, so these are globulin we are generally telling the other name of antibody is the immunoglobulin. So what are the globulin? Globulins are the blood proteins. We have learned about the blood proteins, that the blood plasma is having the protein part, and five types of proteins are there, which we are going to learn in detail into the hematology unit. So out of that five, one protein is the globulin protein. So this antibody is nothing but the blood plasma protein. But why it is called as immunoglobulin? Because it has role into the immune system. So these are modified globulin protein only. So when we talk about the globulin, so blood have three type of globulin. Other globulin proteins are also there in the blood: alpha globulin, beta globulin, gamma globulin, based on their electrophoretic migration rate. So when we talk about the antibody, they are the gamma globulin. These are just classes of the globulin protein. Now, when we talk about the basic structure, so first of all, this basic structure was given by the Potter and Edmund in the year 1950. They said that these antibodies are specified and they are having the Y shape. When we talk about the arm, so here you can see this arm. Arm means the broad Y. So this arm is responsible for the binding of an antigen. Here this red molecule I have suggested as the antigen, and this blue Y-shaped molecule is your antibody. So they said this region, broad arm. Okay, broad arm is responsible for binding to the antigen, and the tail, when we talk about the tail, means this region. This tail is responsible for biological activity, like C activity is the complement activity, or binding to the cells, means it has the various receptors. So on the basis of its basic structure, the antibody, this immunoglobulin are the glycoprotein. It has two type of chain. You can see two chains are there. One is the half build and one is the full length. So this half build or the small chain is called as the light chain, and that heavy polypeptide chain what you can see is called as the heavy chain. Now this heavy and light, these names are given on the because of their molecular weight. When we talk about the light chain, so it is having approximately 25,000 Dalton weight. When we talk about the heavy chain, it is having approximate 50,000 to 70,000 Dalton weight. When we talk about the simplest antibody molecule, so it will have the four polypeptide chain. You can see here 1, 2, 3, 4. Two heavy chains and the two light chain. So the four chains are linked together by the disulfide bond, and an individual antibody always and always consists of two identical H chain and two identical L chains. Now these L chains and these H chains are having the various isotopes. Isotopes means the various forms. So you can see here these are the disulfide bonds are there. This disulfide bond, one must remember disulfide bond are the strongest covalent bond. These disulfide bonds are giving them the proper structure. So proper structure means they are giving them a proper shape, that is the Y shape. But in between this light and heavy chain, other weak interaction can also be seen. Now we know the strong interaction is the covalent bond, and other weak interactions are the hydrogen bond, then salt bridges, ion uh, channels, and all these are the, so likewise the other interaction is also there. Now this disulfide bond can be break up. Now how can we break? By the enzymatic treatment. So when this disulfide bond is broken, this one, when the papain digestion is done, papain is one of the enzyme, so that time this antibody get cuts up from here. So this region, the upper part, which is responsible for the binding of the antigen, is called as the variable region. Why do it called as the variable region? Because each antibody is different from the other. Now, why do it differ? Because it is very, very, very specific and highly designed for the shape of the antigen. So when the antigens are various types, so they also differ in their form. So when we will cut and we will do the papain digestion, so that particular time you will find this Fab and this FC region get broken down. But pepsin when we are using, so this disulfide bridge will be remain, and this FC region. So this region, which is called as the variable region and responsible for the binding of the antigen, is called as the Fab region. And the region which is having the only heavy chains, so this is called as the FC region or the constant region. But when we give the treatment of the mercaptanetol, so that particular time heavy chain and light chains they separate it completely. And same pepsin, when we give treatment, so you will find this disulfide linkage is there, but still FC fragment or two Fab region will be attached. Okay, Fab, though we have seen two are there, so here the two Fab segments are separated. Now, when we talk about light and heavy chains, they are subdivided into the variable and the constant region. Now, what is the variable region? The region which is varying, which is highly specific for the antigen. The region are composed of three-dimensionally folded repeated segment, which is called as the domains. Note down the term domain here. Domains means the particular, the region which is having the specific binding site. So this L chain consists of one VL and one constant, that is CL domain. Most H chain consist of one VH and three constant CH domain. So when you will see the diagram, it will be a bit clear for you. You can see here this variable region. So you can see in the general simple diagram we have seen only this sulfide bond, but it between this CH region and this VH region they also have the disulfide bonds here. So the blue part is the variable region, which is made up of approximate 110 amino acid, and it differs in the all classes of the means all classes of the immunoglobulin. So you can see this variable region is having the VH and the heavy chain, if it is resides, this variable region resides on the heavy chain, it will called as the VH region. If this variable region is reside on the constant region, it will be called as the CH region. But we know the heavy chain is bit longer and it's approximate double than the light chain. So for that reason, this is having the three domains, that is called as the CH1, CH2 and CH3. Likewise, variable region, when we talk about on the light chain, it is having only one variable region and only one constant region. Constant region, till here all the class will have the same antibody. So the variable region of both light and heavy chains have three extremely variable, hypervariable amino acid sequence that is called as the amino terminal end from the antigen binding site. Amino, we know that all the proteins have the two side, one is the amino side and one is the carboxyl side, right? It is because of the amino acid structure. So these proteins are made by the joining of the amino acids. So this is the N terminal, that is called as the amino terminal. So always and always the variable region is present on the amino terminal and the constant region lies on the carboxyl terminal. So when we talk about the isotopes, isotopes means the variable form. In general terms, we are using the class. So five isotopes of antibodies are there, which is called as the IgG, IgM, IgA, IgD, and IgE. So when we talk about the light chains, so light chains, this classification of antibody or the immunoglobulin is done on the basis of heavy chain. The point to be remembered is that the heavy chain in all the class of the antibody differs from each other, and the light chain is having only two types. It can be either kappa or lambda, but it will be identical. It does not mean that one light chain is of kappa or one is of lambda. It is absolutely not like that. We have seen in the basic structure that the chains, the heavy and identical chains, both are always same. So either it will be kappa or it will be the lambda. So on the basis of the amino acid differences into the constant region, both type occur in all class of immunoglobulin. It is not like that kappa is limited to IgG, IgM and the lambda is having so it is absolutely not like that. It is also having that more isoforms. Like if we will talk about the IgG, it is having the subclassification like IgG1, IgG2, IgG3 likewise. Same way we are having the classification in IgGA. So H chains, that is the heavy chains, are distinct for the 5 immunoglobulin classes. On the basis of their only their short abbreviation G, M, A, D, E are given. So when we talk about the IgG, so it is having the gamma type of heavy chain, and when we talk about the IgM, it is having the mu. Likewise, IgA is having the alpha heavy chain, and IgE is having epsilon heavy chain, and IgD is having the delta heavy chain. One by one, we will talk about all the classes of the immunoglobulin. First, we will talk about the IgG because this is something which comes first in our life. That's why we will talk. Now, first comes in our life means this is the only and only antibody which can prevent you when you are in fetus. So, IgG molecule, as I told you, that all the type of antibodies have the 2 light chains and 2 heavy chains which are linked by the disulfide bond. So all the antibody will have one molecular general formula, that is H2L2. H2 means 2 heavy chains, L2 means 2 light chain. So because it has 2 identical antigen binding site, it is said to be the divalent, right? IgG is the predominant antibody in the secondary response and constitute important defense against the bacteria and viruses. That means whenever there is attack of any type of the bacteria or the viruses in the body, so this will be the first antibody. Means when the specific antibody has not been formed, so this will come and this will take care of the viruses. IgG is the only antibody which can cross the placenta. Only its Fc portion have the receptor for the placental cells. That's why it is most abundant immunoglobulin into the newborn babies. IgG is one of the 2 immunoglobulin that can activate the complement system. I hope you will remember complement system is a type of immune response which is responsible for the drilling or punching hole into the bacterial cell wall. So this is one of the dangerous antibody which can activate, and the other is the IgM. So when we talk about the IgG immunoglobulin, it is having the property of opsonization. Opsonization means catch hold, brings up the imprisonment, means just about the locking the movement of the antigen. So this is the structure. You can see 2 antigen binding sites are there. So one antibody is divalent, means one antigen will be caught here and one antigen will be caught here. So IgG, it looks little bit similar with non-secretory IgA. IgA we are going to learn next antibody which is having the 2 forms. One is secretory and one is the non-secretory. So it bit similar, that's why I have put together. So when we talk about the next class of antibody, that is IgA. So IgA is one of the most important immunoglobulin which is present in all type of secretion. When you cry, that time it is present in your ear. When you are sweated, it is present in your sweat. So it is present in your all, means all type of secretion. So this will be the second antibody which is exposed to you when you were new baby, because it is present in abundant quantity in your mother's colostrum. Colostrum is the first milk of the mother. It is also present in your saliva, tear, respiratory, intestinal, genital tract, all type of secretion. It is now the IgA prevents the attachment of microorganism, example bacteria and viruses, to the mucous membrane. That means it will not let anything in. Why it is present into the secretion? Because it is all opening. You will find that usually it is covered with the mucus. Why it is so? Because the mucus contain lots of IgA. So this IgA has a very important role that it cannot allow attachment of any microorganism to that mucous membrane. When we talk about the secretory IgA molecule, it consists of two heavy and two light chain plus one molecule of J. Now what is this J chain? Because this J chain is responsible, it is the secretory component, right? So when it goes into the secretion, it always go as a dimer. So these two antibody molecule are bound together with the help of this J chain, and this secretory component is a polypeptide which is synthesized by the epithelial cells, which provides for IgA passage to the mucosal surface. So means the epithelial cells which bonds epithelial cells of mucous membrane. These are providing the IgA passage to the mucosal surface. That's why it also protects IgA from being degraded into the intestinal tract. As I told you, it is present in all mucous membrane. So we must know that the intestine and other tracts are also having the mucus. So it is having the chances of degradation, because we know that the intestine have the high alkaline pH, and there are chances of degradation of the protein. Now protein degradation, when the protein it changes its pH, and when other factors are affecting the protein, it gets degraded. So this is also made up of protein only. So to prevent its degradation, it is having the special J chain. And when we talk about the serum, some IgA is present into the monomeric form. You can see this is the dimer of IgA. Two molecules are bind, and this is nothing but the J chain. And when we see this is the secretory piece which confers the solubility into the secretion. Next class we will talk is the IgM. IgM is one of the most important immunoglobulin. It comes into the primary response. Primary response means, see, secondary response means the again antigen has exposed. Primary response means this antigen has entered the body first time. So whenever such type of primary responses are there and the invading microorganism is new, IgM will come first, and it is very heavy. So it's just like a percolating tank. To prevent it, it present as a monomer on the surface of virtually B cells, because B cells, all the antibody will present into the monomeric form only. On the surface of the B cell, it will function as an antigen binding receptor. When we talk about the serum it is having the pentamer form. That means it will have the 5H2L2 unit plus one molecule of the J chain. J chain, we know it is very important for the prevention of the degradation. Now when we talk about the pentamer, so it will have the two sides. So 5 into 2, it will have the 10 antigen binding site. Amazing, it can hold at one time the 10 antigens. So it is most efficient immunoglobulin in the agglutination. I had already told you in the previous class that and it forms a complex which is called as the agglutinant. So this also act into the complement fixation, and it have the other antibody reaction. It is very important in defense against bacteria as well as the viruses. It can be produced by the fetus in certain infection. But if it is present into the fetus, that means the baby is having, means the fetus is having some type of the genital or maybe the inborn issues. It has the high avidity of the immunoglobulins. Avidity means the binding of the antigen. Its interaction with antigen can involve 10 of its binding site. That means at one time it can catch hold the 10 antigens. You can see like this. Next is IgE. So IgE, you all remember when we were talking about the mast cells, that the mast cell is having the receptor for the IgE. And what was the role of the mast cell? Hypersensitivity. So this is also having the role into the IgE is having role into the allergy. It that is the hypersensitivity. It mediates or immediate hypersensitivity. It participates in host defense against certain parasites, example helminths and all. Although Ig is present into the trace amount into the normal serum 0.04, but the allergic person will have the great increase amount. IgE may appear into the external secretion. IgE does not fix any complement and it cannot cross the placenta. Next category of the immunoglobulin is IgD. This immunoglobulin has no antibody function, but maybe it can act as antigen receptor. It is present on surface of many B lymphocyte. So it is present in small amount into the serum, but still its any role is not yet definite. So these are the structure: IgG, IgA non-secretory form, IgA secretory form, then IgD, IgE, IgM. Once again, we'll revise. IgG, it is main antibody into the secondary response. Secondary response means the re-exposure of the antigen. We need to remember in the primary response, IgM will take charge first, and in secondary response, the IgG will take charge first. It can do the opsonization. Now opsonization make the easier task for the phagocytic cells to kill, okay, to eat them, because if the bacteria is moving, it is very difficult for the macrophages to run behind them. So they just opsonize, they lock them, and after locking them, it becomes very easy to phagocytes. Then it can fix the complement, which enhances the bacterial killing. We know the complement activation is the punching hole into the bacterial cell wall. It can neutralize bacterial toxins and the viruses. It can cross the placenta. The next, IgA secretory antibody, it can prevent the attachment of the bacteria and viruses to the mucous membrane. It cannot fix the complement. Then one more thing we need to remember, it is present into the dimer form into the secretion and moreover into the serum. Then when we talk about the IgM, primary response, it can fix the complement, it cannot cross the placenta, but it can be formed by the fetus in certain type of the infection. It can be act as the antigen receptor on the B cell, and when it is present on the B cell, it is into the monomeric form, but if it is present into the serum, it is into the pentamer form, and all 5 molecules are joined together by the J chain. IgD, uncertain role, found on the surface of B cell as well as it present into the serum. But when we talk about the IgE, it can mediate the hypersensitivity. It can cause release of the mediators from the mast cell and basophil on exposure of the allergens. It can defend against the parasitic infection which are releasing the enzyme from the eosinophil. It cannot fix complement. It is giving the main host defense against the helminth effect. So this is all about the antibody.